And I worked on the effect of the Francisella pathogenicity island protein, PDPD, on IL-1 beta expression levels in host macrophage cells. So Francisella tularensis is a gram-negative bacterial pathogen. It is also intracellular. So the intracellular part means it just has its life cycle inside the cell. And on the left there is a picture of a macrophage cell being infected by Francisella tularensis. The yellow there is the macrophage cell, and the blue is the Francisella tularensis. So Francisella tularensis is the causative agent of tularemia, which is also known as rabbit fever. There are 4 ways you can contract it. One is through an open cut, and it causes open, painful sores. Another way is through a pore, and it gets under your skin and causes a large abscess, which also is painful. Another way is to get it in your eye, which causes painful redness. And another way is to get it in your lungs, which causes painful death. Yeah, it's also been classified by the US government as a Class A biological agent of bioterrorism and biowarfare. So what it does is it can evade the initial immune response, so it has also been called a stealth pathogen. So how it does this is it enters a cell through phagocytosis, which is basically the macrophage cell kind of going around it and sucking it in through a phagosome, which is like a sac that holds the bacteria. And then after a while it breaks free from this and it starts replicating, and the cell senses this and freaks out and starts sending out all these proteins that signal the immune system called cytokines. And 2 examples are IL-1 beta and TNF-alpha. And this is a diagram of what I just talked about. So as you see there, it's being surrounded by the cell, then it escapes from the phagosome, replicates, and one way the cell can deal with it if it responds fast enough is to surround it with a vacuole, which is the FCV, the Francisella-containing vacuole, and it can just kind of kill the bacteria. Or if it doesn't respond fast enough, the cell goes bye-bye, and the cycle continues. So what the Francisella pathogenicity island is, it's also called the FPI. It is a section of the Francisella genome that contains the coding for genes involved with the pathogenicity of bacteria. And by containing the genes, it also contains the coding for the proteins. And one of the FPI genes is the protein PDPD, and another one is for the transcriptional regulator gene MGLA. So a previous project looked at the vaccine strain of Francisella tularensis, and they found that the bacteria rendered the cells of murine macrophage-like cells, J748.1, incapable of secreting TNF-alpha or IL-1 beta, which means it couldn't respond at all. It was just helpless, in a sense. And then another lab looked at the MGLA knockout strain of the bacteria and looked at the TNF-alpha levels. And they found that the cell could not respond as well without the when the bacteria has the MGLA missing. And then another lab looked at the PDP knockout strain and the same cytokine levels, and they found the same thing. And both of those suggest that the bacteria is phagosomized or hiding out longer because the cell hasn't sensed it yet. So once it breaks free, the cell senses it and sends out cytokines, and if it doesn't sense it, there aren't any cytokines. So the objective of this project is to look at the effect of the FPI protein, PDPD, on cytokine levels, more specifically IL-1 beta, in host macrophage cells. And from this, we hope to gain a better understanding of how Francisella tularensis impacts the immune response in host cells. And so we chose PDP to study because when it was knocked out, it was found to be non-essential to growth or reproduction. And that's important because it can also remain virulent in the cells, which means it can still infect and kill the cells. And we chose the cytokine IL-1 beta because it is activated when the cell senses that the bacteria is present in it. And so my mentor, Lisa Smith, infected the macrophage cells with a subspecies of the bacteria, Francisella tularensis novicida, which was donated by Dr. Frances Nano of the University of Victoria in Victoria, British Columbia. And then after that, she extracted the RNA at 2 hours post-infection, 5 hours post-infection, 12 hours post-infection, and 24 hours post-infection using the Trizol reagent from Invitrogen. And she did it as the manufacturer said to. And then after that, we took the RNA and we reverse transcribed it into cDNA using RT-PCR, reverse transcription polymerase chain reaction, which is basically turning mRNA transcript into synthesized DNA. And so the cDNA levels are directly proportional to the amount of mRNA in the extraction. So more cytokine levels means more mRNA transcript, which means more cDNA. So yeah. Then after that, we took the cDNA from the RT-PCR and used it for the template for the quantitative polymerase chain reaction, which is qT-PCR, in order to quantify the expression levels of the IL-1 beta. And the primers we used targeted the IL-1 beta gene and the beta actin gene. The reason why we use the beta actin gene is because it's present in all eukaryotic cells, which is all cells that are in organisms composed of more than one cell, or grouped together. Okay, and then after that, we analyzed the results we got from the qT-PCR using the 2 to the negative delta delta CT method, which when you analyze data using that method, it expresses the data as a fold change in the expression levels normalized against a internal reference gene, then compared to a control. It's confusing. And so this is an example graph of what the quantitative PCR machine spits out. Again, the DNA levels are proportional to the mRNA transcript. Contrary to what you would believe, the line over here has less DNA in it when it started than the line over here. Because down here is cycle numbers. So with PCR, as we go through each cycle, more DNA is being made, and so that one reached that point sooner, meaning there was more in it. And this is an example graph of what I would like to see. And this is a dissociation curve graph, and what it does is it basically visualizes whether or not there's contamination in it. So it also shows at the point at which the DNA splits into single-stranded DNA, and that is at the peak of each one of these hills here. And each dissociation curve is unique to each DNA molecule. And so what we like to see is one peak per sample set and tight grouping. And if it was a negative control, what we would want to see is a flat line that's tightly grouped across. And this is what I got. So, as you see, the top two are negative controls, and they have peaks. With a negative control, you don't want any DNA in it, and peaks show that there are DNA in it. What I would want to see is more like the bottom half of this graph here, which is what I didn't get, which suggests that there's contamination. And then for these two bottom ones, they are the actual samples. And we don't like these ones because they have multiple peaks for one, like there and there. And there's loose grouping, which suggests contamination again. So this contamination could have been is most likely come from two sources, which is DNA present in the primers and then DNA present in the microfuge tubes post-quantitative PCR. And it's likely a combination of both. And careful lab procedures and new primers will probably fix the problem. And so, my results are inconclusive due to contamination. Yay. And I'd like to thank my PI, Dr. Carson Hoofer, my mentor, Lisa Smith, the RAHI staff, Denise Ortiz, Greg Owens, Renee Pike, and the TCs, except for Matt. The RAHI 2 staff, Dr. Sue Hills and Amanda Meyer. My RAHI 2 instructors, Dr. Adrian Orr and Kathleen Katranis. And thanks to UAF, Alaska BioPrep, Alaska IMBRI, and RAHI for funding and supporting RAHI 2.